speaker today. Let me introduce you to Nate Kirk, who is in the Department of Integrative Biology at Oregon State University. He is a molecular ecologist at OSU and a very talented instructor. He received his bachelor's and master's degree in biology at State University of New York, SUNY at Buffalo, and his PhD in biological sciences at Auburn University. A quote taken from Nate's teaching webpage is, "I believe that students learn best from each other and from practical experience. So I try to limit lecture time in favor of directed learning. Students need a guide as much as a lecturer, and I prefer to act as a bit of both, leading them to make their own discoveries to experience the thrill of science. Therefore, I try to incorporate projects and alternative assessments into my courses." He's designed and implemented several courses, undergraduate courses here at Oregon State University, including these at Hatfield, the Marine Ecology course, the Marine Ecology Laboratory lab, and then the Marine and Estuarine Invertebrate Zoology course. He's taught this last one for nearly 12 years with a focus on student research experience. Utilizing HMSC seawater classrooms and new video technology provided by OSU's Academic Technology, Nate has again redesigned this course, Marine and Estuarine Invert Zoo, as a CURE course at HMSC. So for those of you not familiar with the term CURE, CURE is an acronym for Course-Based Undergraduate Research Experience. It is a novel form of classroom-based courses that offer students hands-on experience doing original research and offers faculty the opportunity to generate new information within their discipline. Without any more delay, please join me in welcoming Dr. Nate Kirk and his students with his presentation.
All right. Thank you for that kind introduction and for coming. I just want to start out with a caveat that this is probably the weirdest talk I've ever put together in my life, but it fits. I mean, this is my home institution. I'd be happy to talk more about science, but I want to talk a little bit about the journey that I've gone on with this course and with these talented students. I also want to highlight everyone up in yellow as a co-author is a student that is either currently taking 461 or took it last year. So there are a number of other students up there as well. I'm a very talented postdoc, Sam Budgood, who I'll highlight some of his work that helped me co-design some of this work that I'm going to talk about. I also wanted to start with, it's not advancing, all that practice. Nope, it's not doing anything. That's okay. Right now. It is online. I feel like there should be some music.
Now try.
Oh, there we go. Okay, thank you. All right, so I just wanted to apologize. Last year I was scheduled to talk, and then this happened the day of my talk. So I was in the hospital with my son, Norm. He is fine and watching at home, so hi, Norm. I'm glad you're feeling better. Anyway, so apologies for last year. When I started teaching this course 12 years ago, I was asked to also give a seminar, and so I did, and that is literally the screenshot from that. And I've been working on a lot of corals, as Yichang mentioned. I still do some coral work. I work on other anemones, and when I started teaching this course, this was the first year. I noticed I didn't have any white hair at that point. I also didn't have any children, so it's correlated, I think. But it was a lot of figuring out stuff on your own. And so Yichang hired me without any sort of experience in teaching a zoology course, an invert course. I'd never even taken an invert course. I just simply worked on inverts. So thank you for taking a chance on me. I was late at night writing the lectures right before I would give them every other day. And the students were just absolutely fantastic. So we went out We did a whole bunch of this. We went out there in the field. Here they are looking for things. We tried to identify everything we could. Yi Chung and lots of people came in to talk about their work, and it was just a really fun experience. And then it sort of culminated as a molecular ecologist. I do a lot of molecular work with phylogenetics. So we would go out there, we would grind up things, and they would try to identify them first, and then we would put them in an evolutionary framework, and then they would present it. We would make phylogenetic mobiles. So if you thought the butcher paper was a pain in the neck, you should see the mobile construction. They convinced me I needed to stop doing that, so we did. Anyway, and we went out there, found a lot of things. The trophy that still exists to whoever gets the most inverts on the BioBlitz, which will happen tomorrow as part of the final exam, and we just had a lot of fun. But it was this month of sort of, I was teaching this course, and this is what I was going to do this month, and that meant that getting papers out and getting more data and things just kind of was on the back burner. And then I always knew about this. I went out. I was so panicked about having to teach natural history out here, having never been to the Oregon coast before. I was a tropical marine ecologist. It was this idea of, I knew there were sea anemones out here. This picture actually went out on a scouting trip to try to identify all these things before I tried to tell other people what these things were. I knew this existed. This is Anthopleura elegantissima. It's the aggregating anemone, is that right? That's the common name? Yeah, aggregating anemone. I learned that a couple of days ago. Anyway, it forms these dense clonal populations. You can tell it's elegantissima by its elegant pink tips that are sticking out of that tentacle right there. And it forms these clones through a really kind of dynamic process of tearing itself in half, which we'll talk about in a second. And it wasn't really until an undergraduate approached me from the University of Minnesota, and they were interested in marine biology, of all things, in the University of Minnesota. And they really wanted to work with somebody marine, but he didn't have a lot of opportunities. And so he asked if I would be his honors college mentor while he would do the work here, and then somebody at the University of Minnesota would help him be his official advisor, and so on and so forth. And I agreed, and we're like, we'll figure something out. And it turned out to be Anthopleura elegantissima. And his work eventually got written up, or is in the process of being written up with some really talented other scientists that helped bring it all together. But it was really sort of a spark of like, oh, okay, I could do this as a side project. And students, when they come to me, will just do the side project because it costs a lot less money to send a student here than to the Caribbean, right? And so it was this idea of these things. And they form these dense colonies through replication. And we know that they defend themselves by fighting one another really aggressively. And to illustrate this is a great video from a colleague at WU, Samantha Lavelle. And this is one of my favorite emails I ever got. The subject was, A, elegantissima is a bully. And so there's my charismatic, elegant one on the left, and Xanthogrammica, this big green one on the right. And this is what they do to each other out there. They inflate these giant things called acaragi, and they swing them, and they leave little bits of them behind. And these are full of syringes called nematocysts that inject neurotoxin. So it's like being in a club fight, like with billy clubs with another person, except there's like syringes of neurotoxin, and you're just bashing each other with that. And eventually, if you force them to, in the literature, it shows that they kill each other after about 5 or 6 days. And so this is a really aggressive interaction, and it beats up on other things. And I wish I could explain. There were some projects that I didn't have time to highlight today that were really cool follow-up data to some of the work that she's been doing at WU. But that video was shot by a student who was in her class that was working on this type of aggression thing. So just even better than anything I've ever shot. So undergraduates contributing forever to my talk. So thank you very much, Camille. Anyway, Point is, we could take these back into the lab, and we know a little bit about their biology. There's supposed division of labor where you have these really intense fighters and then these ones in the middle. We've done a lot of that. I don't believe that argument, but I'll tell you that a different day. But we tried to collect from the middle of the colonies to get the happiest, fattest individuals that we possibly could. And then we can bring them back into the Hatfield seawater tables. We can settle them out into tiles, and they attach quite nicely. And then what's nice about a tile is you can move it around. And so Philip and I kind of worked together, figured out how to move them together. And as you push them together, it's as if they're interacting for the first time, and then you can start doing it. We did 89 fights that term, including plus 10 cell fights, so 100 fights. We lost the pictures for one of the fights. And we were figuring stuff out together. So we were taking pictures. So every 10 minutes, Philip was in there taking a picture of every one of these 100 fights. We were monitoring this for 3 hours. Everybody got monitored for 3 hours. And one went overnight, and he was still monitoring it, still watching it for over 24. I said it's 10, but I think it was over 24 hours. And we were figuring out sort of what these behaviors are. And why I really find this fascinating organism is it has behaviors, but it's not complicated by a central nervous system. There is no response. There's no integration that's happening. Simple nerve nets response is triggered by receptors, and there is no complications of, do I want to do that? Because in behavioral ecology, when you start working on things like vertebrates, they have free will and they do whatever they want, right? And it's usually not helpful. And these don't cooperate enough on their own. So it's really nice to be able to lose that. And so we were moving them together. This was something that he shot on his phone back in the old days of maybe 5 years ago. Okay, well, cutting short a long story, we found an effective size, and that's about it. But we figured out a lot of techniques in this process of learning together. We figured out how we could calculate surface area. We could measure the length of something that we knew, measure in image J the disk of the size, and size correlated. That was the only thing we found. The bigger the size difference, the more likely that was to win. And then, on a scale of run away to stand your ground, those individuals, the size difference, really, the ones that had a bigger size difference, those are the ones that stood around where the little ones ran away. And then if you were small, you were more likely to run, to hide. And so those were sort of some of the behaviors that we'll see, and I'll show you in a second. And we correlated it to a whole bunch of other things that had non-significant features. Really, I just wanted to show this because it also highlights my son out there in the field with my dad. And in the process of looking through and having the slides not be able to advance, I saw that my mom and dad are logged in. Hi, Mom and Dad. Thank you for everything. I love you. And I really appreciate you coming out there with me that day as well. Anyway, not really all that interesting, but it sparked a fire inside of me to how could I use this and start building something. And I love this framework from Sissy Ballin, where we think about cures, course-based undergraduate research experiences in our classrooms as a way of taking a few guided students through a process to how can we possibly mentor 28 students through this process. And what, in this framework, what separates a cure from everything else is at the end of the day, you as the scientist actually care about the answer. And I realized that even when I was teaching it, and people really were getting excited about their phylogenetic relationships of some nudibranch that they found and identified and took really possession of, I didn't care. I mean, I did care. I loved that they were doing that, but it was a lot of time and effort to excite them and get them going, and it didn't help me outside of that satisfaction of helping a student, right? And so how could I then use this and build this into the class so that I could actually get some research done? And there's all sorts of benefits of cures. I'm just going to give you the Reader's Digest. Increased skill sets, self-efficacy, career aspirations, science identity. Especially leads to retention, graduation rates, graduate school enrollments, especially of URM groups. And making a class of 30 in a research, or in some of the stuff we've done on campus, 1,000 students involved in research that's interesting to us, versus the maybe 5 that I could realistically mentor on separate projects. So that's that. So with that, we're very lucky to have some money from NSF to buy Sam out of his postdoc for a little while. And this is Sam Bedgood here. And he worked on Anthopleura as well. And he was really interested in energetics. And he had a couple of papers that just needed a figure or two. And we worked together in co-designing this course last year to answer his questions and generate him some data. So for example, this first plot up here, he was interested in surface area to volume. And I don't want to go into the weeds here because this is his talk and he gave this last year. But basically, the bigger you are, the less surface area you have, and the smaller you are, the more surface area you are. The more surface area you are, that's where the Symbiotidaceae, the algae are. And so that's what allows you to photosynthesize. And anyway, you get the point. He needed some more points for this, especially for elegantissima, because we have down in California where he was doing his PhD, they had much larger elegantissima. And here we have much smaller elegantissima. So he needed some at that far end of that plot. And so that's exactly what we started off. Week 1, there's the protocol. You need to follow it. You're going to do surface area, you're going to do volume, you're going to do measurements. And we have managed to extend out that plot. That is going to be in a publication. And these students are going to get consortia authorship on that publication. Week 2, we wanted it to be a little bit more discovery, yet still in a framework. So we figured this out. I love this picture. It's actually from a paper, Guy et al. 2014. They're supposed to eat anything, right? They're the garbage cans. Anything that falls in their face, they're just going to swallow down and eat. And this is I am not a vertebrate expert, a bird of some sort that fell into the mouth, and here it is being digested, right? And so that's sort of pervasive in the literature that things are being eaten. So what we did was we had students go out there, and this was also repeated in Wu, so we can go to multiple different campuses and do sort of these types of works and experiments. And we gave them all things. Students had to key them out last year using the Light and Smith manual. And then what you're looking at, if it's the left, is all the things that were ingested. It went inside the mouth, inside the gastrovascular cavity. Retained means it didn't just immediately come out. And digested means it stayed, or a shell came out, or some remnant of it. It was actually eaten. And what you can see is the green were things that happened 100% of the time. Blue were things that never happened ever. And yellow were sort of a mixed bag. And you can see that they aren't garbage cans. Only the first third are the things, and things that are incredibly surprising. Pagurus hirsutisculus, the hermit crab, it probably has a common name too. It just crawls out of the gastrovascular cavity like every single time that we have done this assay, and we've now done it 10 times. And it's kind of remarkable, right? I would have never predicted that. And snails, lots of little snails climb out. And then also, interestingly, I thought this was an error, but apparently it's not. Flusterellidae, which is a bryozoan, we found that it was eaten all the time, and Wu found it was never eaten, right? And so there's some interesting things that pop out from those data as well. So that was week 2. I've already used up half my time. I haven't even gotten to the story yet. Okay, so I'm going to pick it up a little bit. We're going to talk about Seal Rock. And Seal Rock is an incredible place. And on the far end of Seal Rock, at the very end, where you see those big rocks down here, I have sort of taken over a rock and filled it with holes, and really found these colonies. And these colonies here are Anthopleura elegantissima that cause themselves to fight with each other and causes those big breaks that we see between these colonies. And I'll trace that colony for you to make it a little bit easier to see. You can see there's 1, 2, 3 colonies next to each other of all clonal individuals in between. And we know that they fight over this space, again, to the death if you force them to. But my questions are sort of, I'm here, and we can go out here, and I'm probably going to keep teaching this course. What's this rock going to look like in 5, 10, 20 years? I don't think I'll be here much longer than 20 years, but you can make some hypotheses, right? And so aggression, if we can measure aggression in the lab, what's the point of being small and forever boxed in by your neighbors when there seems like there's real estate above and below, and you could just beat up your neighbor and take their space, right? And so what's causing that? Sex and aggression, sex is really interesting in this, because there's a number of different ways that it can occur. It can occur asexually, where they literally tear themselves in half. Okay, we can bring them into the lab and we can watch them rip themselves in half. In fact, we have some, if you're curious, I could show you some that are in the process of tearing themselves apart. They leave a scar, they're not able to eat. It's really annoying for feeding assays when they start dividing and you're trying to feed them, right? But that is what it is. And here's one, oh, it's not advancing. Here's where it's tearing at the very top, and then that's the same individuals the next day where it's ripped itself and now it's next to each other. But they also, in addition to fighting, they're also fighting their sexual partners because these clones are sexually generated. They will go out as planula larvae, recruit back, and that is a little bit not understood of how they recruit. And then at that point, they can either make eggs or sperm, and that's how I'm going to define male and female for this talk, is the capability of making eggs. On the right, you can see eggs under a light scope and the nuclei under a compound scope. And then on the left, you have the male. Okay, so we think it wouldn't make a lot of sense to be beating up on your sexual partner. So maybe you would take it easy on a compatible sexual partner, but you would be more aggressive towards your own sex. Anyway, things that we can test. So we went out there, marked these colonies. Here's my rock at the far end. And here are all our colonies, okay? And we've labeled them out here. And they're along about a 20-meter transect at the far end here. And along this transect, there are all these colonies. And here's colony 39, for example. And we know 39 last year had 973 individuals, thanks to Jean. Jean was wonderful. She went through and looked at all of these pictures and told me how many animals were in each one of them. So I am forever grateful for Jean for that. We also know that it's female. We know that its position in the tidal height based on a laser level that we went out and measured. We know it takes up about 2 meters of space out on the intertidal. And we have been fighting it. So we're starting to get a better sense of its aggressiveness and competitive ability, but more on that. And we went out there and we marked them with Z-spar. And I could tell you that this lasted about a week. And in about a week from last year, we have one marker that remains. It's kind of fascinating. So with my field tech Norman, we went out and we drilled into the rock, and there's colony number 11 that he's pointing to right here. So thank you for helping me with that. And fascinating things, sort of an even sex ratio, and I'll talk more about that in a little bit. Maybe I'm running out of time. Look down at the far bottom. So this is arranged by sex. We can see that there's a big cluster of males, okay? And the most logical thing is that these males down here are all the same clone, and it's just been split up. But what I can tell you is that we have been following these colonies for time, and they look pretty much the same with boundaries with each other. And if they were the same clone, if you split and grew into it, wouldn't you just merge? Okay. So we have been collecting genetic data. So I'm a molecular ecologist. So we're going to do some 2B rad genotyping to see if all of these are separate or if all they're the same. But at the moment, that is an open question. And there tends to be clusters of female colonies over here to the far left, right? And then some really interesting unknowns where we don't know the sex. And that is, you can dissect them open and look at the sex. And it's not just from lack of trying. We've looked at 10, 15 individuals, and they're all asexual. And we did it this year, and it's the same. So there are some that are forgoing sexual reproduction for whatever reason, right? They're dividing, in my opinion, too fast. But fast forward, what can we use this class for? This year, everybody recognizes colony 13 in the first couple of rows because they counted every individual in there. All 27 students. And we came up with an average of 89.2 plus or minus 3 individuals, okay? And so I now have an estimate of error that I can use, okay? So I plan on doing this a couple times to get a better sense of our error rates that we're getting from our pictures of being able to do this. But we can follow this colony. So there it is in spring. We went out there with a different course, and we went out there and measured it again, and had 134 individuals that time. And the summer, we now see it has 168 individuals. Okay, so over time, but notice 14, okay, so 12 off to the left. We can barely see 12 here, okay? It has receded a ton where it used to be like kind of right up along it. It has receded quite a bit. And here we can just barely see, it should almost be touching, but it's not. and it is retracted quite a way, except for that little bit that is touching, while 13 keeps getting bigger. And 14 has shrunk. Okay. So again, if these are a clone, I would expect them to merge together, and they have not. I can go out there and I'll be like, yep, that's 13, that's 14, so on and so forth. This is the worst figure ever. My students are laughing at me because I made it because I thought it would be helpful to us. And then as soon as I made it, I'd be like, this sucks. Like, we can't use this. But we've been slowly filling this. This is my fight matrix of everybody against everybody. And this is the progress we've made in just 2 years. Okay. So eventually I want to show the aggression towards everybody and have replication of that, but we're a ways out. Okay. One of the really interesting projects that came out last year was really nice work that I co-designed with Elise Tisdale. I'll talk in a second about her work. But she was really curious in this course about what happens when you repetitively fight them, because they don't just fight once and then they're locked next to each other unless they detach and get washed away. And so we can bring them back into the lab. We can settle them out on these things, keep them at bay, and then Yi-Chung masterfully managed to have video camera feeds. So instead of taking a picture every 10 minutes for hours, we figured out that we could figure out and score their aggression in the first hour of the fight. So we've been cutting it down to about an hour of video footage, but we can collect 8 fights at once, video footage from 8 fights at once. Okay, and so let's talk about a little bit about how to score the data, and I'm going to turn it over to some students to talk about their work and their results. So you can have no acaragi out. Those are the fighting clubs that you beat each other and inject each other with neurotoxins. You can have them facing away from your competitor. Why you would do that? Maybe you were fighting somebody else and you just haven't extended it towards your competitor. You can have it facing your competitor, or you can have it totally full. Okay, so these are the categories that we're going to start with. And then we let them bash each other. And I want to show you some of the behaviors that we've come across as aggressive responses. So this is something called an acaragi sweep. All right, so you can practice an acaragi sweep yourself. You can lift up your acaragi and you can bring it down. It's exactly what it sounds like. So let's watch. So the one on the right is bringing it down and bashing number 48. All right, so it's really taking it out on number 48. You can retract in. So if you're getting beat up, you're not thinking, oh man, I'm being beat up, I should get out of here. You can just simply release water from your hydrostatic skeleton, and you can retract down. And the one on the right, I believe, is left is going to do that. All right, so you can protect yourself because you're not out and about. You can run away. And over time, we can start to see when they do it, and we can see who runs away first, who gets bashed the most, who is retracted most of the time, and we can get a really good sense of who's winning and who's losing, and who's more aggressive and who's less aggressive. So fast forward to Elise's work. I'm really proud of this work that she has done. She put in a tremendous amount of work, presented this last year at Western Society of Naturalists, and there also got a job offer. She's currently working Hi, Elise. I think you're also zooming in. So I hope I got that story right. The first thing we looked at is the number of tentacle hits. So how many times they flinched when they came in contact with each other. And what she found was really interesting. Here's fight 1, fight 2, and fight 4. So as it went on, as you fought them more and more, they got more touchy. Okay, they got more aggressive. And then we gave them a break, about a 5-day break between fight 4 and fight 5. It would fight consecutive days. And then it reset. Okay, it went back down to the same level as sort of fight 1. Okay, and then this happened in acaragi sweeps as well. You could see them going up and then down. It wasn't significant, but it's the same pattern. Okay, and then this is really fascinating, Dave. The minute to the first runaway, when the first one decides it's had enough. Fight 1 took a while. Fight 2, a little bit less time. Fight 4, less time. Fight 5, they booked it. However, if you look at the sample sizes on the top, in the first fight, it took them a while to run away, but they all did. Fight 2, only half of them ran away, and then less and less and less. So if they decided they were going to book it, and there's no decisions, right? There's no central nervous system. They booked it immediately. Otherwise, they stood and fought. Okay, oh, you again. Let's do it. Right, and that was kind of a more response, and it was really interesting. And then the acaragi status, whether they out. At first, nobody had them. These were from the middle of the colonies. They weren't interacting. We hadn't stressed them too badly in the lab. They were fat and happy, ready. At the end of the fight, man, they had inflated their acaragi. They were willing to fight. And what she found was that it retained it. Some of them reset back to having no acaragi out, but some of them kept them up. Okay, and so basically we've taken these reproductives and forced them to become warriors by interacting them with one another. And one of the very fascinating things, which I mean, I just collected some of these data yesterday. There seems to be an effective colony. So certain colonies are better than other colonies, and so that is extremely interesting to me. So without further ado, I want to turn it over to Jake and Kaden, who followed up on this experiment, and here's their data.
Awesome. So this is our acaragi status at the beginning of our fights for all 5 days of our experiment. And for the most part, most anemones had no acaragi inflated at the beginning of the fights. But surprisingly, there was always a couple that had them inflated. And this is to compare with the end of all fights for all 5 days. And surprisingly, not all anemones at the end of their fights had acaragi inflated. In fact, less than half of them did. And so this was really interesting. That kind of is interesting for our question. We expected a higher amount of acaragi inflation. And it's really important for our question to note that acaragi status is not a direct indicator of aggression. It's more of a proxy for aggression, which helps to answer our question.
So we also looked at tentacle hits of each of the fights. So we have data for fight 1, fight 4, and fight 5 right now. And as you can see, when you look at the graph itself, it doesn't have a specifically clear trend like Elisa's data did, which we found very interesting. It does go down towards the ends of the fights, but it doesn't reset like we saw in other data sets. When we ran it through R, we also found no statistical results or significant results when we went through the two variables between number of tentacle hits and which fight they were going through. For acaragi sweeps, we did the same thing. When we looked at them, you can again see that it does go down, but there isn't a super clear trend, which we found very interesting. If you can see that on the left graph here, just with the average number of sweeps, and you can see that there isn't a clear distinction of either increasing or decreasing as the fights went on. And on the graph on the right-hand side there, it's average minutes to first sweep. So the first time that we noted that either of them swept in the fight. And again, it doesn't match what we found last year. So when we ran it through R, we also found that there wasn't a statistical significance between the two variables of those two as well. And finally, we looked at aggressive responses. So for time spent retracted, you can see that as the fights went on, it slightly went up and then went down a little bit again when you hit day 5 after that break. But again, it didn't come back statistically significant, and it doesn't have a strong trend line either. Average minutes to first run had the strongest one from the preliminary data that you can see, which you can see when you look at it from fight 1, it took a very long time for them to run, and then it went drastically down and decreased in time by the time it hit fight 4 and fight 5. That being said, though, when we did run it through R, we again found no statistical results between those two variables as well.
You should always publish immediately before you repeat it with higher sample size, because you're always going to get the opposite pattern of what you expect. And that's exactly what we got. We got the complete opposite pattern. That said, they randomly, I mean, they literally had a week to do this, so 2 weeks. So that said, we only looked at 8 of the 36 fights that we've done. So there is still that, but this will be, we'll make it a larger thing. So hopefully a story will emerge, and maybe even the one that we think will emerge. So there's other things out here that I'm interested in. I talked about sex ratios. One of the fascinating things in the literature is the sex ratios, if you look through, are incredibly weird and skewed. There's some papers that have like no males, 8% males, and others that are 100% males at certain sites. Some of it is sampling size, right? Looking at very small numbers and you're finding these, but these skewed ratios are not uncommonly reported. And so we were, I had a student a couple of years ago that was a Ursa Engage student that was really interested in that sex ratios and like, was that true at Seal Rock? And I could say that our rock, no. Okay, they're not. They're about an equal sex ratio out on our rock. But again, there's clustering of the males and clustering of the females. So if we stopped only looking at the bottom part, we would be like, oh yeah, everything's a male, right? And that's weird. So what Bailey did, they went through and they went to all these different rocks and looked at it. And so, oh man, oh males. This was really male dominated, our site. So we had almost 2/3 males if you go across all of Seal Rock, including a few from our rock. Okay, and you can see the sample size and the pie charts on there, and who's next to who. We could do a quick and dirty test, and we can see that unlike on our rock, where it was significantly likely to find the same sex next to you, here it was p-value of 0.07. So again, you should never repeat your stuff. You should always just publish it in case you get something like that. But anyway, this sex, there's a lot of really cool biology that comes about it. And with that, I'm going to turn it over to Miranda to talk about some really cool sex data that they uncovered this year. Tyler and Miranda.
Hello. In my experiment, I was looking at tidal position and how it affects gonadal status. On the graph, you can see there's a bunch of colors. 0 represents there are no identifiable gonads, and 5 represents that there are fully developed gonads. And we can see that in 2022, there were not a lot of zeros compared to 2023, and in 2022 there are a lot of 5 gonadal statuses compared to 2023. And we found that there was no statistical significance between gonad status and tidal position, but there was a statistical significance in the difference in gonad status between the years of 2022 and 2023.
When we start looking at it, gonadal status goes from sort of 0 on a scale from Brian Bingham's lab up to just kind of exploding out gonads. And so when you sex them, you can see just tons and tons of them. And we're seeing lots of highly developed gonads last year, and this year not so much. Okay, and that's that. And so does sex actually matter? My hypothesis I gave you a little bit earlier was whether or not you would be more likely to be kind to a sexual partner than a competitor. And to help explain some of those data, I'm going to turn it over to Erica. And oh, sorry, Erica and Bianca. Yes.
So when we started looking at the data, I was interested in which sex would win more often when we fought them. Is it males or is it females? But Erica also came up with a really good question of, is it more aggressive of a fight when you're fighting the same sexes or when you're fighting opposite sexes, which is a lot of what Nate just talked about. So we went ahead and during our experiment, we paired males with males, females with females, and males with females. And so along this x-axis, you can see along the bottom that we've labeled what type of fight it was. And then along that y-axis, you can see that in this particular case, we measured aggressiveness with aggregation hits, but flinches or time retracted could also be interesting things to measure. And so in the future, we likely would measure those. But in this case, we decided to measure it with aggregation hits. So I'll hand it off to Erica to kind of explain the trends.
And so after we ran our fights, we analyzed the results to come up with this graph. And we observed that there wasn't much aggression in male-female or male-male fights. But however, we noticed a very interesting trend in the female-female fights seem to be much more aggressive than the other two categories, and they were enacting a lot more aggregation sweeps than the other two. And we know that some other classmates of ours found similar trends in this data, so it would be interesting to see if this pattern repeats itself with the larger sample size in the future.
Okay, and it's such an inherently cool thing. And I've been told multiple times by people in my lab group and others that I'm barking up the wrong tree. It's broadcast spawner. It doesn't matter if you're right next to each other versus this. And so a little bit of indication in case you're on Zoom. But as I was thinking about this, I didn't remember until today that this is hanging in the academic wing. SERM, which is an outstanding class where through Fish and Wildlife, where people come and do a really intensive week out here. Is it a week out here? Do I have that right? Okay. And then they go do independent research projects and they come back and do a poster. I got it right. Okay. Kyle approached me last year wanting to work with me, but he was stuck in a hospital guarding a door for long periods of time. And he couldn't stop guarding the door to come out here and do experiments, but he needed a project to do. And so we had all this video fighting footage, and I said, okay, why don't you pick a topic and a variable and we'll look at it together. And I'll be darned, he picked the same thing. Just dawned on me today. And this was hanging up there. And I'll be darned, females were striking more than males were striking each other. Males were taking it easy in both sets. These were completely different colonies because we've been purposely avoiding fights in the same. So for whatever our reason, our males in our data set are less likely to strike out than females. And his p-value 0.057. I know p-values aren't anything I'm really focusing on, but that's what pilot data does, right? It messes with your p-values. But anyway, I still think there's something there, and I think some students will follow up on this. But I'm going to turn it over to Lacey and Brad to talk about other cool things.
All right, so for our project, we were looking at how colonies were changing between 2022 and 2023 and their aggression. But while we were collecting data, we found that there was a trend with female colonies shrinking, both in surface area and change in individuals. So this is with colony sex on the x-axis and change in individuals by percent, change in surface area. And in both cases, females were shrinking and males were growing. And yeah. That was about it.
Okay, so this was surprising. So first of all, what I was most terrified of was that we were going to find out that every colony was shrinking by 5 individuals, which was the number we collected last year. So here's this incredibly static thing, and here I am out there killing it, 5 individuals at a time. But that's not at all what we found. We found some individuals that were increasing and some that were losing hundreds of individual, ceding almost a ½ meter of space out there. Okay, and some colonies, one colony is gone. 27 is where it's supposed to be, 28 is where it's supposed to be, 26 is not there. Okay. And so what is, I have samples, like I have genetic samples. What is with 26? I don't know. And here's 40 here. You can see that there's 935 of them. This year there was 735 of them. We have lost 200 individuals on this. Interestingly, I plotted this. These are data I made this graph about 20 minutes before I'm talking today. So change in the number of individuals on the x-axis and change in the surface area here, it's highly correlated. So as we lose individuals, we're losing colony space. And so counting in this, that's going to be something that we're then going to be able to correlate. We tried in this class, but we only have 17 out of our 50 colonies, this data. And then as we fill in how aggressive these colonies are, we would predict that the more aggressive you are, the more likely you are to stay out there. And I will tell you that females are incredibly aggressive. But with that said, I'm going to turn it over to Daphne, Daria, and Sean to talk about division in the colonies, asexual reproduction, as well as sexual reproduction. And you have to stand in this blue box, or else Zoomland can't see you.
Okay. So for our experiment, we were looking to see if there would be a difference in percent division between each of the colonies. So what we were looking at is we took the samples, had them back in the lab, and then we counted how many individuals were dividing on a certain day, and we wrote that all down. And then we divided it into high dividing and low dividing. So anything over 0.5 is considered a high dividing colony, and anything less than that is low.
Yeah, and so if we can move to the next slide.
Perfect. I'm just going to briefly kind of talk about our summarized data where we saw kind of an interesting trend that I'll get to after my little blurb here. And so on the x-axis, you can see we have marked the number of divisions out of 5 individuals as we collected 5 individuals from each colony. And on the y-axis, you can see gonadal status and development ranging from 0 to 5, 0 being no discernible gonad development, and 5 being very discernible and noticeable gonad development. I won't get into the methods of things, but basically, yeah, we separated the trends by sex. So you can see the male trend line is in orange and the female trend line is in blue. And we got some interesting results showing significant statistical differences between the two sexes and their divisions in gonad status. And the main point being that, as you can see with that blue trend line, females had a more aggressive slope, meaning that the more they divided, the higher their gonad status was. And that was pretty contradictory of our initial hypothesis, but that's science. And as a class, I'm sure we're all very thankful for Nate for allowing us to explore these different avenues. So yeah.
So this was completely counter to what we had predicted in terms of resource allocation and things along those lines. And again, sex rearing its head in an unexpected way. I would never predict that there'd be differences here in this. And then I just want to follow up as I run out of time on a couple of things. Like some of these, as I mentioned at the beginning, why are some of these colonies always asexual? So this is a colony that has added about 50 individuals over time. We have sexed probably about 15 individuals from this colony. So even us taking that many individuals, it's still managed to increase by that much. It is divider, and we never find gonads in it. Even in these big ones in the middle that are certainly past the threshold of size, that there's a size cutoff before you can develop gonads. They just never do. Okay. And then there are some colonies that are teeny tiny out there and some that are massive and big, okay, where the individuals are all big and some that are teeny tiny, which means they have some sort of genetic thing that says divide, 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 where others are slow dividers. And it turns out yesterday, I was talking to Will Ryan, who does this work in Dianemone, a different sea anemone that doesn't hang out with its clones after it divides. And yeah, there are totally fast dividers and low dividers. So I think we're going to collaborate together and get some more students involved and do some of this stuff next year. Anyway, some take-home thoughts. There are just so many impressive starts, so many interesting directions that I'm going to take this research next year. And they all got to see each other's present similar trends. We started to see like there's something weird about females and males. This whether you make eggs or sperm, there's something different about you. Whether it's fighting, whether it's you, and you would expect these females, when they're super aggressive and winning all these fights in the lab, they're the ones that are shrinking out there on the rocks. So we haven't again, this is data that's coming in this week, so it's hard to put our heads around it. And the genetics will help out sort some of these questions, but that's that. We have started a long-term monitoring. Now that those screws out there, we have permission from the state for 5 years. Hopefully in 5 years, they'll let me continue to do that and help us get those demographic study. In exchange, I can offer authorship as part of these courses and experience in skills. Going forward, this talk is only going to get more interesting unless they fire me, right? So this is 1 year of data. I was expecting no changes or very little changes, and we're seeing hundreds of individuals added or lost from colonies. And I was not expecting that. We have set up an additional site. We have gone down to Yachats. In fact, we were there this morning sexing individuals until about noon, when I probably should have been writing this talk. But anyway, that's that. Well, we have genetic samples from all these different places, different years for rad genotyping, and that's that. We have some RNA-seq data that actually has come back from the sequencing companies. And so we're going to be able to look at this outside warrior versus reproductive stuff. So we'll be able to put that in Philip's paper. And continue pairing and aggression. Some students are actually, the course is pretty much over. They're going to go back to the lab with me right now and pair some animals that we just got from Yachats, some males and some females, and see if we can, what it's like down there, because it's very different. And I just can't tell you how grateful I am for the opportunity to have taught this class to these students who presented their own work. And I just highlighted some of it. There was fantastic work all across the board, and things like feeding and fighting against different things that have given us all sorts of ideas and directions to pursue. And hopefully some students will continue to work on these projects throughout the year. And also we have a lot of digital data. And I plan on bringing it into my eCampus courses so that people can start to do original research. Because before I was like, oh, I'm sorry, I really don't have a lot of projects unless you're really good at R or you're really good at programming, right? But now I've got these videos and these protocols, and so it's great. So I'm just truly indebted to the 2022, 2023 students for being troopers. As we've, every time you take something that works perfectly well and then you break it, there are a lot of cracks in it. So thank you for putting up with us. Yichang, I'm grateful for the opportunity and for continuing to back our research by getting us cameras, getting us videos, getting us support. Ed for helping us figure out how to do that, Cinnamon for inviting me and dealing with me, and really everybody. I see a lot of people out in the community that I'm just grateful for. I'm also grateful that we had funding to buy out SAM and some other things. I agreed. I offered Kate an opportunity to make a plug to Orsi, and this is the slide she came up with. So I volunteered. It's my fault. She said I could modify it. I was tempted to put your picture in there instead. But Tracy and Kate have done a fantastic job. I do urge you to look it out. And I just wanted to end. I don't see John. Is John here? John is not here today. I just wanted to highlight John Chapman, a special thank you to John. Those of you that don't know John, John is a incredible natural history and invasive species biologist. And when I was here 13 years ago and didn't know anything, I learned a lot from John. And John, one of the more enjoyable days I had at Hatfield was I spent half a day at him just walking around collecting things for a genomics project that we both got roped into. And he was just like picking up this crazy isopod and just barely looking at it and be like, this is some species you've never heard of. I'm like, oh, okay. And it took me like hours to key them out later. But anyway, the point is, when he gave me some really sage advice off the cuff. He just said, whenever you get your permanent position, whatever you do, start monitoring it, because in your lifetime, it's going to change. And I tell every single one of my students the exact same thing. I wasn't expecting it to be in 1 year, but we have started monitoring. So I hope I'm still going to do my DNA stuff, but I hope in some small way that I'm helping the cause the task you set me to. So thank you, John. And with that, I'd be happy to take any questions.
Any questions from the room? From online? Oh.
Great talk. Just
thank you.
Fantastic work. I think if I heard you and understood you correctly, you said that there was a difference in the number of or in the sexual development between 1 year and the other. Was that correct?
Yes.
Did you look at any cofactors like productivity, ocean productivity or something? And I'm just wondering if that, if you looked at that, that seems, like it might be of interest.
Absolutely. So all the student data I found out about yesterday. So that is exactly right. And the obvious thing is in the news stories, they're screaming about the hottest July ever. And so that is an obvious thing. And so temperature in the lunar cycle are gonna tie into reproduction and when they spawn. So it's possible that they've already spawned, or it's possible that they're stressed and they're reabsorbing them. And so there's no tissues in there. There's no, they can store fats, but they're usually in balls in their mesenteries where their gonads develop. And so in the coral literature, when they get stressed, they reabsorb their gametes. So I would believe either one of those. And so we could have when I go out here in the spring for a spring course, we absolutely should be sampling them and sexing them and teaching them how to do that in that course. And then when they come out to this course, they're already trained in that, right? And they can explore something else or really set to task. So I absolutely. Miranda and Tyler, that was one of their interests too, was temperature. But yeah, I have, there's most of the literature ends in 1983. There's just a ton of behavioral, a lot of things about Anthopleura. And then for whatever reason, 1983, it just peters out. And there's some wonderful people still working on it, but I would love collaborators. So if I've convinced any of you to work on Anthopleura, that's awesome. Let's talk, especially if you have expertises in metadata and how to correlate it to things that we're seeing in our data. That's great.
Any other questions?
Look to me like the individuals at the perimeter of a colony were the ones doing the fighting. What role do the ones in the interior of the colony play? Are they relevant?
Yeah, absolutely. So there is a literature on a division of labor. And so the outside are called warriors, and the inside are called reproductives. And there's some scouts and individuals that go out and get beat up and then they come back and so on and so forth. And there's a number of papers. They actually physically brought this massive boulder into the lab and they followed what was happening real time as they were interacting with each other. That's where we started. And really what we were finding was size trumped all. And where do you get big? In the middle, where you're happy. You're surrounded in the immersion stress when the water disappears. You are out there in the dry. And if you're surrounded by wet individuals that are your friends, you're going to do much better. There's higher temperatures on the outside of the individuals. They're also the ones fighting. They're the ones fighting off predators. They're the ones interacting with they're doing all the crap work. And so basically we can make that phenotype just simply by bringing it into the lab and stressing it. And one thing that I would have presented these data, but they're not significant. We fought some repetitively over and over. We're stressing the crap out of these poor animals. They don't have gonads at the end. And so I just need a because we went back to the tanks and out in the field and they still have gonads. So I think we're stressing them and making warriors because we're fighting them. But yes, there is a trade-off of that, and it's probably energy. And then we also did last year, somebody was really interested in the sex of the warriors versus the sex, the status, the gonadal status, and there was no significant difference, despite being in the literature that there's way more in the reproductives in the middle. But yeah, great question.
I had a quick question for you, Nate.
Cinnamon.
So at the Seal Rock site, is anybody doing long-term water quality monitoring at that site that you could correlate with some of these changes?
Oh. Is that true, Sarah? Do you know of anybody? Okay. Yeah. Recently, I don't remember who, but a student ran up to me and be like, "Have you looked at the news today?" And we were like out there, and apparently it's like, "Oh, is that what all that brown is?" Like, I just thought it was some sort of weird thing. And yeah, I don't know. I will say this. Initially, I was pretty pissed at the permitting. I'm sorry, I think I should watch what I'm saying. Thank you for permitting me. Like, I had asked for Boiler Bay because we had historical data there, and that was pretty much our go-to for a site. And they denied me and said, well, you should look at Seal Rock instead. And I was like, the tourist destination? It's gonna be like, we're gonna be overrun with tourists all the time. But I've come to love that because my students do the work and I run interference and get to talk to a lot of people about sea anemones and what we're doing. And they're like, what are these screws for? And I get to explain like why we felt necessary to modify the habitat. And I've come to love it, but Yachats is going to be a little bit less run with people. And so I'm hoping that we can still find a couple of sites. It turns out that monitoring this one rock is a ton of work. And unless I get like 18 new graduate students and have the department support them, I'm pretty stretched. And so my rock is going to be my rock, and I'm going to set up some satellite things to test. But again, partners, I would love partners if people are interested, especially if we can involve students, because students are the great partners.
Can I ask one more little question?
Absolutely.
Do we know the average age of an individual?
That is a fantastic question. So the big Anthopleura elegantissima, there are some back of the napkin calculations that they could be 200 years old. But if I was to measure your growth rate right now, I would measure you to be extraordinarily old too, right? And so there's some problems. And if you stuff the faces of these, they can grow. And so the question is, how good of an indication of that is it truly? I can refer you to some great papers for that. But the age-old question of how old is a clone? Because they divide, and then is it just born, or are they both 2 years old, or so on? And so how long are some of these colonies been here? There's also some back of the napkin sort of math that has been done to calculate like the division rates in a given colony and how many individuals are out there. But what we're noticing, and Sam actually could probably speak to this too if he was here, is that when you start to isolate them, they divide, they get lonely. And I would hypothesize that there is some sort of contact that keeps you from dividing more. And if you're alone, you're going to divide more. But it's also energetics, right? And so you tend to get more food if you're in a group. I don't know. It is the short answer, but it's a great question.
Thank you.
Yeah, you bet.
All right. One more question.
Not a question, but I just looked up. Surfrider does, I think their weekly water quality and
Oh, thank you.
Pterococcus measurements, and they do Seal Rock, Hill Creek, ocean, and tributary samples.
Cool.
So you can take a look at.
Sounds like a collaboration there.
Yeah, there you go.
All right. That concludes our seminar today. Thank you for joining us, and please thank you. Join me in thanking Nate and his class for presenting. Us online as well. Can we get the class to come up for a group photo?